Welcome back friends. Welcome back in another video tutorial for microbiology with Shagni. So in this video tutorial, I am going to talk with you about RNA. RNA in details. Although here we will not going to talk about the types of RNAs in details, but here I will be focusing on the RNA from the point of view of biochemistry. That means here we will going to talk about the properties of the RNA molecule. Okay, so this video will going to be the video for RNA from the point of view of biochemistry, and then in some other videos we will going to talk about this RNA from the point of view of molecular biology also. There we will going to talk about tRNA, rRNA and mRNA in details, but it will be in some other videos. So let's start today's discussion. So what is RNA molecule? The full form of RNA molecule is ribonucleic acid, right? So from where it come? It come from DNA molecule by the process called transcription, right? So remember one thing, that the DNA molecule which is in the nucleus is the genetic material of us, right? But this DNA will never leave the nucleus, right? Instead, DNA will be transcribed into RNA, and this RNA will going to leave from the nucleus and go in the ribosome and produce protein, right? And that's how RNA transfers genetic information during protein synthesis, right? So you may understand how much important this RNA is. If RNA was not there, then from the DNA, the genetic information cannot be transferred to the protein, right? That means it acts as a bridge between DNA and protein, right? But it is not the sole function of RNA. There are various RNA that was discovered later that also show catalytic activity. That means there are some RNA present who have the ability to act as an enzyme, and we will call those RNA as ribozyme. Let me change the color so that you can visualize it very well. I think this color will be better. So, so we have learned that RNA can act as a transfer of genetic information during protein synthesis, as well as some RNA have catalytic activity. Okay, now in some viruses, this RNA directly can act as a genetic material, meaning instead of DNA, in those viruses, there will be RNA who act as a genetic material. Okay, for example, tobacco mosaic virus, and here we will going to see the experiment, that is the reconstitutional experiment of H. Frankel Conrad and B. Singer, that help us to understand RNA as genetic material in the later part of this video. So now, until now, we have understood that RNA can act as a transfer of genetic information during protein synthesis, as well as it have catalytic activity like protein, and also some RNA can act as a genetic material, right? Now let's move on. We will going to classify RNA molecule. So there are two classification of RNA. One is genetic RNA, where RNA directly acting as a genetic material, and another type of RNAs are non-genetic RNA. So we can divide non-genetic RNA into further two parts, coding RNA and non-coding RNA. The example of coding RNA is mRNA, and non-coding RNA example is tRNA, rRNA, snRNA, snORNA, etc. etc. There are many more. Okay, so this is the first thing. Now let's move on to the thermodynamic stability of RNA secondary structure. Before that, just like DNA, the primary structure of RNA is nothing but the sequence of the nucleotides. But remember here one thing, here the ribose group have 2' OH group, okay, which was not present in case of DNA. That's why we are calling those type of ribose as deoxyribose, right? But here we will call them as ribose, right? So that's the idea, and that is a very significant difference between DNA and RNA. Okay, we will going to learn about it in details in the later part of this video. Now, the secondary structure of RNA is due to, just like DNA, Watson-Crick base pairing. Okay, that is the canonical base pairing. Here, A will going to interact with U, because we all know that in the place of thymine, there is uracil in DNA, sorry, in RNA, and guanine will going to pair with cytosine. There will be 2 hydrogen bond between adenine and uracil in RNA and 3 hydrogen bond between guanine and cytosine both in DNA and RNA, right? So that's how by canonical or Watson-Crick base pairing the RNA double helix will be created. And very very importantly, the RNA double helix will be shorter than DNA double helix. Okay. Now let's move to a difference between RNA double helix and DNA double helix. Remember, RNA double helix adopt A conformation and DNA double helix, which is normal DNA double helix in physiological condition, adopt B conformation. Now what is the AB? What is this? So in the DNA, we will going to learn about the types of DNA molecule, where I will going to teach you about A DNA, B DNA, Z DNA. There you will going to learn about the A conformation, B conformation, right? So for now, just understand that RNA double helix follow the A conformation and DNA double helix follow the B conformation. Okay. Remember another important thing, that RNA double helix, which follows A conformation, is more stable than DNA double helix. And the RNA double helix required a high temperature for its denaturation, but DNA double helix require low temperature as compared to the temperature required in RNA double helix. Okay, what is the reason behind it? Why RNA double helix require a high amount of temperature for denaturation than DNA double helix? So the idea is more base stacking energy, okay, which is observed in A conformation than B conformation. That's the idea. So the RNA double helix is shorter than DNA double helix. RNA double helix adopt A conformation, DNA double helix adopt B conformation. RNA double helix is more stable in terms of denaturation by applying temperature, and DNA double helix in that case less stable. But let me clear that if we going to hydrolyze RNA molecule or RNA double helix or anything with the help of alkali condition, then RNA hydrolysis will be much easier than DNA. Okay, because DNA lacks 2' OH group. In 2' position of DNA, there is simple H, and that provides DNA an advantage during the alkaline condition. In alkaline condition, DNA is more stable than RNA. Okay, so now, so here another thing you should remember, that the secondary structure of RNA is more stable and the formation of secondary structure favors the folding of RNA. Okay, we have studied about another type of folding in biomolecules, that is the protein folding, right? And in protein folding, we have studied that tertiary structure favors protein folding, right? But in case of RNA, it is the secondary structure. Although here also tertiary structure forms in case of RNA, but those type of tertiary structures are very weak. Okay, the stable form of the RNA is the secondary structure, but tertiary structure also forms, and it will be via weak interaction between secondary structure of RNA. Clear? So that's the thing. So here we have studied about the secondary structure of RNA. Now let's move to the alkali catalyzed cleavage of RNA. So this is very very important thing from the point of view of RNA chapter. You may get a number of questions from that topic. So try to understand it very very carefully. What will going to happen if we going to hydrolyze RNA molecule under alkaline condition? So hydroxide ion will come, and hydroxide ion will going to attack the 2' OH group. Okay, and as a result, what will happen? There will be formation of 2' alkoxide ion. Okay, and this 2' alkoxide ion will going to attack the 3' phosphate group. Okay, the phosphate group which is present at the 3' end, and as a result, what will happen? This phosphoanhydride bond will going to broken, and that's how there will be generation of a cyclic 2 prime 3 prime nucleoside monophosphate. So sorry for one thing, this bond is the phosphodiester bond, not phosphoanhydride bond. So sorry for this thing. And that's the idea. So that's how a transesterification reaction will occur, and we will get cyclic 2 prime 3 prime nucleoside monophosphate. So what is going on here? In alkaline condition, if we going to hydrolyze the RNA molecule, hydroxide ion will going to attack the 2 prime OH group, and this attack creates 2 prime alkoxide ion, and this 2 prime alkoxide ion will going to attack the 3 prime phosphodiester group, and that's how the phosphodiester bond here will going to be broken down. Okay, now it will create cyclic 2 prime 3 prime nucleoside monophosphate and the remaining part of the RNA chain. Okay, now the thing is not over here. The another hydroxide ion will again going to react this 2 prime 3 prime cyclic monophosphate, leads to the formation of 2 prime nucleoside monophosphate and 3 prime nucleoside monophosphate. That's how under alkaline condition hydrolysis of RNA will going to occur. So if we going to discuss it again, so hydroxide ion will going to come first, and it will going to attack the 2 prime OH group, right, generating 2 prime alkoxide ion. Now this 2 prime alkoxide ion will going to attack the 3 prime phosphodiester group, leading to the cleavage of phosphodiester bond and formation of cyclic 2 prime 3 prime nucleoside monophosphate and the rest part of the RNA molecule. Now this monophosphate will going to again hydrolyze by another OH group, which will going to act on the 2 prime 3 prime monophosphate, leading to the formation of 2 prime nucleoside monophosphate and 3 prime nucleoside monophosphate. That's how it is done in case of RNA. But this reaction will not going to occur in case of DNA, because remember from where it all begins, that is the 2 prime OH group is absent in DNA, and that's why DNA is more stable under alkaline condition than RNA. Remember it very carefully. Okay, you may face various type of MCQ from that topic, so be prepared for that. So that is the alkali catalyzed cleavage of RNA. Now another important thing and interesting thing, that is the RNA world hypothesis, because there was a question that is going on in scientists' mind, that is, what was the first self-replicating molecule in our earth? Because try to think about DNA requires primer for its replication, right? So DNA need a protein for its synthesis, right? Similarly, protein needs RNA molecule, right? If protein do not get mRNA molecule or not protein, if ribosome not get mRNA molecule, then nor ribosome nor tRNA will going to facilitate the formation of protein, right? That means the synthesis of protein requires RNA. Similarly, the synthesis of DNA requires primer, which is a protein, right, or which is coming from protein, right? That is the primase protein, right? That simply suggests that nor DNA nor protein can exist independently, right? So the biomolecules that remain is RNA. And during the discussion of biochemical properties, we have observed that RNA can both act as a genetic material, right? Not only that, it can store, transmit, and double genetic information. And these both characteristics is also observed in DNA, right? Not only that, this RNA can also have catalytic activity, right, which is enzyme like activity. That means it can also do things that a protein can do, right? That means RNA can both act as, or can both function as DNA as well as protein, right? I mean to say that the functions of DNA and the function of protein can both be done by RNA molecule, right? So on the basis of that simple observation, RNA world hypothesis was proposed. So the phrase RNA world was first used by Walter Gilbert in the year 1986. What was the hypothesis according to RNA world? RNA world hypothesis suggested that RNA was the first self-replicating molecule in the earth. Later, a cell membrane was formed surrounding it, leading to the formation of prokaryotic cell. That is the idea of RNA world hypothesis. That simply suggests that RNA was the first living thing in the earth. Later, a cell membrane developed surrounding it, leading to the formation of prokaryotic cell. And what is the basis of RNA world hypothesis? RNA world hypothesis is based on the idea that RNA can act as a catalytic molecule. Also, it can have the ability to transmit genetic information. That means the properties of both DNA as well as protein. So that is the idea of RNA world hypothesis. Okay, so I hope it is very clear to you now. So now let's move to another topic, that is RNA as a genetic material. So as I have just told you that some virus contain RNA as genetic material, so it was proposed by it was proved by an experiment, that is the reconstitution experiment of H. Frankel, Conrad and B. Singer. So what they have done, they have extracted the protein from TMB type A. So let me start it from the very basic. What they have done, they have taken two TMB virus strain. What is TMB? TMB is tobacco mosaic virus, right? They have taken two TMB virus strain, and we all know that virus generally consist of two things: protein and DNA or RNA, right? So this TMB virus contains RNA as their genetic material. So what they have done, they have isolated both both RNA and protein from both the strain. Okay, and they have created a hybrid RNA by taking the type A protein and type B RNA once, and also they have created another hybrid virus by taking the type A RNA and type B protein. And what they have done, they have used that hybrid RNA to infect tobacco leaf. Now, surprisingly, what they have observed that the lesion type in the tobacco leaf is depending upon what type of RNA they have used, or from the RNA or the RNA they have used belongs to which strain. Okay, for example, in this subset of experiment, they have observed that the lesion in the tobacco leaf is similar to the lesion caused by TMB type B. And why is this so? Is this because we have used TMB type B for the generation of this hybrid virus, right? Similarly, in that experiment, they have observed that the lesion in the TMB tobacco leaf is due to the presence of TMB type A RNA, right? So that simply suggests that RNA is the genetic material, because the type of RNA which is present in that hybrid virus, the lesion is going to occur. Okay, so that is the idea of reconstitutional experiment by H. Frankel Conrad and B. Singer, and that simply proved that RNA can also act as a genetic material. So that is the video for our RNA. So here we have covered a various type of things. We have learned about the thermodynamic stability of RNA molecule. We have learned the alkali catalyzed cleavage of RNA, RNA world hypothesis, and at last RNA has genetic material. So I hope this video will going to be helpful for you. Thank you.